start understanding the kind of work that we do. And let us start with Indumathi, because I understand Indumathi's work is at a more lab level, and you are not only working at the cutting edge of gene editing tools, but also stem cell technologies. So, what does both these kinds of tools offer us today? How do they come together? What kinds of questions are you addressing using those? If you could tell us a little bit.
So to introduce myself, I work in an eye hospital. So we deal with problems of eye disorders, particularly if there is a degenerative conditions, whether we can regenerate these tissues for therapeutic purposes. Similarly, now we have latest technology, but embryos, you have to rely on embryos which are ethical. So now we have a latest technology where you can convert any cells in the body to behave like embryonic stem cells. So this is possible by a Nobel-winning technology called iPSCs, which is induced pluripotent stem cells, where people have been trying to understand what makes embryonic stem cells pluripotent. There are many genes that are involved in the process, and people have come up with a small list of 3 or 4 genes which are important. When you put in these genes in any somatic cells, say it is a blood cell or a skin cell, you can convert it into embryonic-like. So that technology has really made given a lot of promise for many tissues that cannot be regenerated by existing adult stem cells in the body. So we employ the technology to see if we can make retinal cells and see if we can regenerate these tissues. So this is possible. I mean, if you take a healthy cells from any human body, then you can convert it into iPSCs. So once they reach the iPSC stage, you can differentiate them. I mean, you can teach the cells how to make retinal cells in the dish. So that's what we do. So we call it as a stem cell therapy, but it is a stem cell derived retinal cell therapy. Okay, so this is possible if it is a healthy tissue, right? You make healthy cells, but if it is a patient who have genetic condition. So the genetic makeup is the same, whether it is a skin cell or eye cells, the genetic makeup is the same. If there is a mutation in the eye, there is a mutation in the skin as well. So if you make iPSCs, you have mutations in the iPSCs as well. So how do we make autologous cell therapy with patients' own cells? How do we provide a cell therapy for the same patient? So that is where the latest technology like CRISPR editing has come in, where we make iPSCs of the patients and then use this CRISPR technology to edit and correct. So once you correct it in the stem cells, you can then make cells which can be tailored for the patient. So that is broadly the area that we work in the lab to develop cell-based therapies for the patients. And there's also another angle in the sense that there are over several genes, 300, 400 genes are implicated in the disease, but we don't understand how each of these genes cause the disease. So there's called disease modeling studies we also do in the lab, where we try and create such patient-specific cells with mutations, but we don't know why this gene is causing the disease. We try and understand that using this as a model system in the lab, understand what is the biology there, how the disease is manifested, if there is a way of intervening and creating a therapy for these. So that's broadly what we do in the lab, and this can be extrapolated for any diseases of the body, any inherited diseases of the body. Say it is a muscle disorder or the blood disorder, the broad principle is the same. So that's in summary of what we do and what stem cell and gene editing therapy means.
Thank you very much. So if I might summarize it, you're looking at eye diseases and you're looking at degenerative diseases of eye, where the cells are going bad, and you're finding stem cells of eye, changing genes in there and putting it back into the eye. And are these human eyes? Human cells. And okay. And are you able to put it back into the human eye, or is this mice models? Or
so right now it is at the experimental stage, but there are clinical trials already started around the world. There are several groups in Japan, US, and UK are working in this, and the trials have already started. And it's shown that the cells that are derived from these stem cells can be used in therapy, and they are safe, provided the process with which we make them or safer, and there are critical regulatory guidelines, critical parameters that we need to check in. If that meets the criteria, then it is totally feasible to clinically translate.
Got it. Thank you. Arkasubhra, would you like to now explain us the work that you do?
Right. So, I'm a molecular biologist, and in our lab, we are essentially trying to understand how diseases work. And when I say how diseases work, we are trying to understand how genes cause diseases, or mutations in genes cause diseases. So we are what our genes make us, right? So everybody knows that. We look at like our parents, grandparents, uncles, etc., because we inherit those genes. But as we go through life, we can accrue some changes in those genes. And all of us have those changes. Every generation has those changes. But every once in a while, you'll get a change which causes some of those proteins to not work, and therefore create a disease. So at the base of it, gene therapy is very logical. You have a problem in your genes, you create a drug that fixes the gene. So that is essentially gene therapy. So any kind of DNA, RNA, or nucleic acid based therapy, the process of delivering it to the cells of which are affected, be it, say, if it's blindness, it's the eyes; if you have a neurological condition, it's the brain; if it's muscular dystrophy, it's the muscles. So that is called gene therapy. So it's the corrected gene, or the good copy of the gene, and the delivery process together makes a gene therapy. So that's essentially what we are trying to do in case of gene therapy. And of course, we also use the same sort of tools that Indu talked about. So where we take a patient's own cells and create the type of cell that we want to correct the mutation in. So it's like, let's say you have a chair here, right? So this is of a certain shape and everything, but it's made from plastic. Yes. You take the cells from the skin or blood cells, you sort of turn back the clock and make it into a stem cell, and now you can guide that cell to become an eye cell, a neuronal cell, or a muscle cell. So we do those to check if we can fix the gene for that particular patient. And then we have to test it in animal models, etc. So this is a story that nature took note of. A couple of years ago, a family approached us, and they had a 19-year-old girl who had this ultra rare condition called phenip, accumulation of bad proteins, and that kills the neurons there. So this girl, unfortunately, was born with a mutation in a certain gene called SERPINI1, and that caused the SERPINI1 protein to aggregate. The neurons were not able to clear it out, and this protein aggregated and started killing her cortex. So at the time when she came to us, although she was a 19-year-old, she had the cognitive ability of maybe a 2-year-old. Okay, so she was fine, top of her class till like 7th or 8th grade. Then she started having seizures. So it took almost 4 years for people to identify what had happened to her, which was done at NYU, at New York University, is where they did a genetic test. And then they figured out that this was a mutation and not just a seizure disease or something else. Right? So they weren't able to treat her properly. So at that point, there was no other option for treatment. So the only therapy for these kinds of conditions is a genetic therapy. So that's when they approached Devajyoti and me. So we created, so Devajyoti has these gene editing enzymes. So our genes are made out of only 4 letters, and they are in a sequence that can be read by the protein machinery in the cell. So now you can create a synthetic protein machinery that reads the same thing, but it directs it to a certain point where you can correct that single letter Just like on a typewriter that you create a new, you can, put a white mark and put a new one there. It's just like that. You can delete and create a new sequence there. So that's gene editing for you. But then the challenge was how to get it to the brain, right? So you can do it on a dish. That's very relatively simpler to do, but actually doing it in a patient requires that you figure out how to get it to the brain. So we did a lot of studies where we injected the therapy in the bloodstream, inside the brain, through the spinal cord, and we also had to look at whether it was safe. So when you inject it at different places, the efficiency of that therapy to reach the brain is different. And then the immune response, the patient's bad response to it is also different. So we understood all of that, we created that. Now remember, this is an ultra rare condition that this girl had. She was one of only 3 known cases around the world. This is the only one in India. So therefore, it is very difficult to solve problems for such cases. The second thing, of course, was before we test anything on humans, we test it on animals, on lab rats. And they were quite open to the idea of doing a N of 1 trial. So an N of 1 trial means it's an ultra rare condition, only that patient has it. So from a regulatory standpoint, the reason they make you jump through so many hoops is because they want to know who is liable if something goes wrong. Entire onus of liability has to be mitigated by doing stepwise trials on many different kinds of animals, doing lots of different tests. Yes, it's a pain to do, but it has to be done to ensure that we have done all our due diligence to make it safe for human use. So we got through that process very quickly. However, the patient passed away before we could give her the therapy.
Does that also mean that each of these regulatory things will need to be done for each single patient because these are such personalized treatments?
Yeah, that's a great question. Actually, yes. But what around the world people are doing now and are working towards is getting approvals for the platform. So like the gene editing that we will do, like in this case this was a base editor. This same editor, just by changing its target, can be used for many different patients, many different diseases. So now if the main platform is approved, then you may not need to do all of those things each time for each patient.
So Indu, I know that your PhD was in basic sciences, so maybe I would like to understand from you how you saw this field evolve in the country.
So I was trained as a cell biologist and a molecular biologist, not really a stem cell biology. So my foray into stem cell research came in when there was a problem presented to me, like there is a retinal condition and cell therapy is the only option, right? Cell or gene therapy. So what the projects that you formulate is to apply the technology that you learned as a cell biologist. So basically stem cells is also a cell, right? So it's just the behavior of the cell is different from any other normal adult somatic cells. But the technology or the process of culturing, growing the cells is all the same, but what each cell needs is different. So we just understood that and we started applying these principles and started developing the program of stem cell program for different conditions like for cornea and retina, and so you can do the same thing for any other tissues as well. So you really don't have to be a stem cell expert to start a stem cell research. You have to be a good cell biologist and a molecular biologist. And similarly, the technology of gene editing, which Arka talked about, it's also a simple molecular technology. So it's not, the gene editing or the CRISPR editing is not very new, though it has made into a big application in the recent times, in the recent 5-10 years, but it's been known since 1980s. So these are mechanisms that small organisms like bacteria and even older evolutionary particularly older bacteria like archaebacteria, routinely employs to protect themselves against invading viruses. Okay, the viruses that infect bacteria are called bacteriophages. So when they infect, they inject their genome into the bacteria. So bacteria has devised this mechanism of combating this infection by attacking, recognizing the viral genome, binding to it and cutting and chewing off and eliminating further infection. This is like an adaptive immunity we develop in the body, like when there is an infection. The vaccines also work based on that. So you introduce the antigen, our immune cells recognize it and keeps it as a memory. The second time you get the infection, you get to combat it. Same mechanisms bacteria have used. This is a very old evolutionary conserved mechanism bacteria have evolved. Now the application, once the scientists have understood, this has been understood starting from our 1985 onwards. There were publications on this, and around 2010 or 2012, people have realized that this technology can be now harnessed for targeting not just viral genomes, any DNA or RNA. So our DNA RNA can be human gene or bacterial gene or a plant gene. Anything can be targeted. So the mechanism works the same. So for understanding this and applying this technology, you need to understand how the mechanism works, and you have to be a good molecular biologist to make this happen. So that's how the system, the technology has been adapted, and.
Are you meaning to say that we are still using the bacterial CRISPR-Cas9 system into other animals, humans included?
Yes. So it's been used in the regular gut bacteria. That's E. coli we routinely use in the lab. E. coli is the workhorse in the lab. And we also have, it's been reported in Streptococcus, strep throat.
What I meant to ask is, for example, when Arkasubhra built the therapy for this young girl, did you use the CRISPR-Cas9 system of a bacteria to actually change the genes of?
Exactly.
Originated from a bacteria, but we make it undergo what's called protein evolution. So using molecular biology tools, we use different aspects of different proteins that we like. We evolve that to make it very specific and applicable in mammalian systems, in human systems.
Got it. And from there, Indu, I will move to Arkasubhra because you work in a much more hospital-like setup, if I understand it correctly.
I mean, we both work in similar setups.
Yeah. No, I mean, I know Indu did her PhD from CCMB, and I'm familiar with the fundamental biology nature of the place. So what I want to understand is how was, I mean, if we go back in history, if we go back in history, how do you think these hospital setups adopted the at some point of time, very basic molecular biology tools for themselves? I'm also asking this because generally we also see this gap between the tools that are being used or developed in fundamental biology labs reaching a hospital space.
Right. So the use of molecular tools in hospital setups has come today because of genetics. So that's one primary thing that hospitals have been using is genetic testing. But even before that, molecular tools that assay for specific proteins, right? Cancer. Today, if you go to if a patient is to be diagnosed with cancer, there's an array of molecular tests that are done, from your biopsy samples, from your blood samples, and of course the genetic tests. So there is a lot of movement towards molecular testing that is there in hospitals. In our hospital in particular, the whole aim of setting up the research arm was to equip clinicians with additional knowledge or additional tests that can better stratify the patient. So if you go and say, for something even as simple as, say, a laser eye correction surgery to get rid of your glasses, now you have a dozen different technologies which are available for it. But which would be the most suitable for you? Which would, for you, give the best outcome on day one and not cause, let's say, a dry eye or glares or halos when you're driving at night. So these are considerations for quality of life, which a molecular tool can give you prediction for. So that's why hospitals like ours are interested in that, because we are moving more and more towards personalized medicines, bespoke therapy, etc. I mean, if you take a poll of everybody that's sitting here today, if you get a headache, most of us will take, say, one medicine, but there will be a few who will need to take 2 pills to get rid of it, and there will be some who can just sleep it off. They don't even need to take a pill, right? So unless molecular testing comes in, a clinician won't be able to differentiate that. So that's why, molecular tests, molecular readouts are coming into all fields of science. And the third thing, of course, is drugs. Modern era drugs, like antibody drugs, they are against specific targets. So like diabetic retinopathy, we give anti-VEGFs, right? So for a variety of different, say, inflammatory conditions, we give, let's say psoriasis, you give an anti-CD6. These are antibodies against specific targets that are floating around in your body. So for a clinician, knowing that is important. How much dose to give, whether I should continue with this drug or should I switch over to a different drug. So that's why molecular testing.
No, and you remind me very well that, I mean, so far we have been talking about gene editing only in terms of therapies, but
right.
It's also very, very useful in terms of diagnostics.
Absolutely,
and both are equally important when we are thinking about treating and managing diseases.
Yes.
So that also now compels me to ask, how prevalent are these being uptaken by hospitals? What kinds of facilities do we now see in hospitals? Are these very far off and few, or this is becoming more and more mainstream?
Right. So this sort of ties into your earlier question that you asked about uptake in India and availability of the technical know-how, etc. So, of course, India and the global south in general lags behind the Western counterparts. I mean, the amount of awareness of such kind of tests and such kind of diagnostics or therapies is higher amongst both the general population as well as the clinical clinicians. Here it is lower. And part of that is because what you are doing today, this is not a mainstream activity. So if you were to go to Japan, for example, in the morning news, there will be a section on science always. They'll talk about iPSCs and stem cells and gene editing and everything. So everybody knows about it. But here it's not so common. So that's a major issue. So therefore uptake is slower. The second thing, of course, is the infrastructure requirement that is there to initiate these things. Initially, everything costs a lot more unless volume kicks in and then prices start dropping. So when genetic testing first came, it used to cost 70,000 per sample. Now you can get it done for 10,000 or even 7,000 rupees. So it took 10 years to come down to that level. So therefore, in India, everything is slower. So in all these bigger urban centers, if you go to most of the big cancer hospitals, eye hospitals, etc., they have the infrastructure already. They have people who can do molecular testing, genetic testing. Your molecular diagnostic labs have now proliferated quite well. So PCR-based diagnoses are very, very common now for a variety of different viral things, bacterial things, etc. So definitely there will be uptake. The strength that we have is that we take a long time to move. But once we move, then it snowballs very quickly and becomes a very large sort of activity very, very fast, because we have the population that needs it.
So talking of the snowball, I'm reminded of the very recent Genome India project that we have been seeing in the media. What was this project about? If Indumathi can tell us a little bit on that, and also what kinds of opportunities a project like these open up for people like you.
So the Genome India project aims to sequence the human genome, particularly Indian genomes. At least in the phase 1, it aims to sequence at least 10,000 human genomes of Indian patients spread across the country or all 4 zones, and use that information and to understand the genetic variability in the population in the country, and see if that can help us to understand the disease prevalence in the country. This is the first phase, aiming to sequence the normal individuals. What is a normal genetic variation in the population? In the second phase, it also aims to capture populations with specific genetic conditions, diseases, and understand what is the spectrum of disease mutation that is prevailing in the country, because what is a mutation that is more common in India may be different in the West, and we can't really apply the same technology. So that's the idea about the Genome India project. So once that information is curated and is available for the scientific community, it will allow us to develop many gene-based therapies, and we know which is the most prevalent mutation, because in case of gene therapy, we are designing therapy for the gene, for the sequence-specific therapies are being designed, for which we need that information. So what is lacking at this moment for this to be broadly taken up is we don't even know which diseases are inherited, what are the genes involved, what are the spectrum of mutation that's available and more common in the country. So that information, though many of our research labs are in isolation pocket doing gene sequencing, and there are sequencing companies that are doing it, but all that information is not available in a public domain for us to make a policy decision on that. So that is what is majorly lacking, and hopefully this Genome India project will help us in a big way. That.
Would you like to add anything to that, Arka?
Right. I mean, whatever she said, the one thing that I would like to point out is that the link between genetics, environment, and diet. So the Genome India project gives you that first part of it, the blueprint. What was our blueprint? Because one of the things that we've suffered from particularly in diseases, for example, in age-related eye diseases, it's quite well known now that there are certain mutations in the West which is prevalent, against which people are making drugs, against which we are also making drugs, trying to make drugs here in India. But those mutations are not prevalent in our Indian population. They are different. So our patients actually need a different type of drug or a different design of gene therapy or a cell therapy. So that hopefully Genome India project would enable. I think the bigger aspect of this, so these sorts of work has happened in the past. You know, you have the Genome Asia project that came out in 2019, which also had Indian genomes in it, and other South Asian genomes and linkages between them. I think the important thing here is the size of the data. We all talk about AI, right? So an AI-based drug design based on genetic sequences is only going to be good if you have a very large database.
How large does it need to be?
Given our population, you should have at least 10% of it, right? So, 160 million, right?
Yeah.
So we should have at least 16 million. 10,000 is just a drop in the bucket. 10,000 is not enough, but at least it's a start.
And even without the Genome India project, what are the other diseases that the community looks at as priority right now to deal with using these kinds of technologies?
So I think even in the Genome India project, they have listed the top 3, which includes cardiovascular diseases and hematological diseases. And I think neurodegenerative conditions are top on the list.
Is that because those diseases are very prevalent, or we know more about those genes?
Yes, it's because they are more prevalent, and also, so in terms of numbers, it's hard to comment that way, because even the rare diseases in Indian population, rare is not really rare. And even the conditions that Arka was talked about, the FENEB is the only patient, he said. It's because that's the only patient who has sequence and we know the information. It's just that we don't even know, and the genetic testing is not so prevalent in the country. Like even the clinicians are not totally informed about the genetic spectrum, the disease spectrum that is available, and need to advise and counsel the patients to go for gene therapy. But that's picking up now. We have genetic counselors being placed in many major hospitals and being recommended. So they do identify inherited conditions. So if there is a consanguinity in the families, they do advise them to go for genetic testing. Those information are now available in pockets, but hopefully we really need a system to compile all this information. When the COVID hit, ICMR was getting the information on COVID positive thing on a real-time basis, right? Every day basis, they were updating the database. That was made possible because all these diagnostic laboratories were registered with ICMR and they were compelled to upload this information. Thereby, the government is able to compile those information. But that's not the case in case of genetic disorder. We do a lot of genetic testing, but we don't have the information in one place. Probably an initiative to coordinate all this information and have a database will really make a big difference to also have an idea about which is the prevalent disorder, which are the particular genes that are involved for a specific conditions. So that big information is missing, and when it is feasible to do it during COVID in an urgent basis, I'm sure this is also possible. I think the initiative should be started in that direction.
Thank you. I'll go back to the FENUP case and diseases like these, which are rare and ultra rare and are extremely expensive to treat. I mean, do we see more conversations happening towards treatments of such diseases, which is kind of the opposite of cardiovascular diseases, unless we find such ultra rare mutations there too?
Right. So, before I answer your question, so rare disease, the concept of it is obviously a misnomer for India, right? So the numbers today, as per census and epidemiological studies in the United States, not in India, is that there are 30 million people in the United States who fall under the genetic disease category, who have a pathological problem because of mutations in their genes. So if you consider the population of the United States and in India, we are 6 times of it. So we would have somewhere between 180 million to 200 million people in India who have these. Every single person sitting here, knowingly or unknowingly, knows some rare disease patients. We probably don't identify them because of what she said. The genetic testing is not so prevalent. The doctors are not as aware. So they are not identified, and either they die or live through life with a disability without knowing that it was a genetic disease to begin with. So now comes the question of bespoke therapies. So bespoke therapies, the reason this FENUP case was highlighted by Nature was because this was a very unique technology. There were 2 unique technologies which, if we would have treated, would have been first in the world. It's not first in India, would have been first in the world. So the first one was the type of editing that we were doing. It was a base editing that we were doing. There are no base editing technology trials that have happened anywhere in the world. Second was this particular therapy was being done with this vector called AAV vector. So this is the gold standard of doing gene therapy. However, there is a limitation of AAV vectors in terms of the cargo that it can carry, how much genetic material it can carry. So our lab is one of the few labs in the world that works on this unique sort of cutting and pasting technology, right? We take a large gene which cannot fit into AAV, we cut it into 2 pieces, put one piece in one AAV vector, another piece in another AAV vector, then transduce the patient with both vectors. Inside the patient cell, these 2 pieces come together, join and make the whole gene again. So it does work, but it's a new tech, it works in animal models, etc. So this trial would have been the first for both of those. Therefore, the development of it was expensive, but that expense was still, I would say, less than $500,000, right? Usually, to develop a gene therapy in the United States, it takes somewhere between 70 to $100 million. Here in India, we are able to do it $500,000, although every single instrument and reagent that we buy is the same that the Americans are buying. On top of it, we are paying extra GST. Right? So it is more expensive to buy here in India. And on top of it, it takes 3 months for anything to come to India, right? There you can buy it that evening, right? You just make a request, it's there that evening or next morning. So despite those, we are able to do. Now the point of how it is expensive. So, and if you look at bone marrow transplants that are available today, right, you're not so bothered about, a bone marrow transplant. It's still going to cost you 15-20 lakhs, right? If you look at a heart transplant, the entire process of that can probably be 60-80 lakhs. So a gene therapy being in that same ballpark of 10, maybe 5-10 lakhs for an eye disease, to a systemic disease like sickle cell anemia being 50 lakhs, or cancer being 40 lakhs, or a muscle disease being 1 crore, is still within the same ballpark of other diseases. So we are not that far off, because we are not selling the 30 crore, 50 crore kind of therapies which are available in the West at the same thing here. The entire plot of why we are doing indigenous technology development, why we want people to come back to India with these technologies, set up their shop here and try and build it, is to make it accessible. And if we hold the IPs here, then it is accessible. A bulk of the cost in the West is not just the cost of goods, it's the cost of the IP.
So I will ask the last question before we open up for the audience now. So talking of, I'll move a little bit from diseases, because every now and then in newspapers we see we have found a gene for this, a gene for that. And now some of these genes could be relevant as well. Like if I could find a gene that can get rid of common cold for me forever, that will be wonderful. I think so personally. And then there are genes, of course, of many other intangible things that we also see. And with the development of these technologies, there are also these businesses coming up, right? Which offer everything from designer babies to designer dogs. So, as experts in the field, how do you look at it? Are there ethical concerns that we should be keeping in mind? And anything that you would like to advise, both of you?
So the technology is available. It can be used for certain conditions. But the choice of where you want to apply really matters. Like if it is a condition which can help improve the life of a patient, say a patient cannot see and you're giving a life for the patient, the patient's liver cannot function, I'm going to improve the liver function. That is ethical to do. But if you're thinking about changing the color of the individual, I'm going to make a superhuman by engineering the muscle mass of an individual, or a super sports person, then there is an ethical concern there, right? So whether it is required is the question like all scientists have to ask. If it is important to do and if it's going to change the life of an individual, it is allowed as per the ethics. And also with respect to cell therapy or gene editing therapy, we have something called somatic cell therapy and embryonic editing. Okay, somatic means any adult's tissue you can modify, because this is done with the consent of the patient, right? So patient agrees that I'm going to take this therapy and we are allowed to do, and your regulations allows you to do, and it stays with you. If I'm editing a patient with an eye condition and correcting the mutation, it benefits that patient, it doesn't pass on to the next generation. Okay, so but the other aspect is embryonic editing. Embryonic in the sense like we have a problem, inherited condition, it's running in the family, but we don't want the next next generation to be affected. So can you edit and correct in the germline? So that's something called germline editing or the embryo editing, which is not allowed ethically also. And there is a global moratorium on that, as scientific community has agreed in consensus, and they've established a moratorium that embryo editing is not allowed in humans. Reason why is the ethics involved is the consenting, right? So the future generation cannot consent for what you do today. So it's legally not correct and ethically incorrect to put your next generation into a risk which they really don't want. So you anticipate with respect to the outcome, you have the expected outcome and unanticipated outcomes, right? So the anticipated and unanticipated. You don't know today. You do edit a gene, and you say that it has a function in the eye, and you edit and correct it. What if the same gene has function elsewhere in other tissues? Okay, so these things we don't know as of now. So we cannot do such kind of embryological editing at this point. So the guidelines clearly state that so you can only do somatic editing and not germline editing as of now.
Right, just to extend on that, when we are developing these therapies, we have to prove that it is not, there's no vertical transmission, meaning the patient that got treated, it doesn't affect their eggs or sperm, so they don't transfer it to the next generation. So, ethics is what she told about. But at the same time, it has to be not just the law, but people following the law and understanding why the law is there. That's most important. These are extremely powerful techniques, right? So in our lab, we have tools that can literally do anything you ask for. Yeah. So in certain countries, there are black market gene doping clinics. So, for example, athletes can go get bigger muscles, stronger muscles, more stamina. They can run faster, jump higher, and these can't get caught by general dope tests. So, live longer. Lots of people are trying to do how to live longer, but repeatedly, current level of science has shown that you use any of these technologies that can make a cell live longer are the same technologies which will cause cancer, because cancer cells are immortal. So you can live on as cancer, you can become immortal as a cancer cell, of course, but if you want to live longer, you're going to get more of these diseases as well. So it's a double-edged sword.
Thank you, Indumathi and Arkasubhra, for the questions so far, and we will now open up for the audience.
Two points that you made. One was about the cost of gene editing, very high. And the second thing is your self-imposed guidelines of not doing what I will call cosmetic gene editing, okay? How do you reconcile these two? To bring the cost down, you probably will need to have more gene editing rather than less gene editing, right? What are your views on this?
Right. So, bringing the cost down is a question of accessibility of the technology. So that is our primary goal, where we maintain quality but keep the costs relevant so that more people can access it. And of course, with current level of resources that are available, it cannot go below that. So gene editing or gene therapy technologies will forever remain as very targeted bespoke therapies. That's how it's meant to be. But how we produce the therapy might get improved, which is what will bring the cost down. And your second question is more about adoption of the technology by a wider swath of people. So yes, having sort of cosmetic things in gene editing will definitely make people more aware and more the fear of using the technology will go off. It's like laser eye surgery things, right? When it first came 20 years ago, 25 years ago, people were, I don't want a laser in my eye, come on, use a blade or something. But now everybody does it, and it's not a big deal anymore, right? Even though it is inexpensive, it costs like 1 lakh almost, right? So still it is quite acceptable. But I think the moment you go to cosmetic applications of it, these techniques are just so very powerful that drawing those lines is very, very important. If those lines are blurry, say for example, it is extremely easy to mutate pathogens, right? That's what she was talking about. You do it and it's very easy to design them in a way where it affects a large swath of people very, it's not that expensive. So you want to have a certain barrier to the technology and to the price so that it doesn't become that people can do it in their garage. So I think that should be one practical barrier for these sorts of powerful technologies, where you don't make it so simple that a high school student can do it. So if the high school student's girlfriend says that, I want fairer skin, and you make a quick edit of your melanin genes and put it in as a cream, it will work. But we don't know whether that will lead to other side effects later. So you need to have some of those barriers, I think.
So if I can add a different angle to this. So I would want to talk about the scalability aspect of it, right? So for conditions, there are certain conditions which are common in many patients. So in that case, the moment you have a process that can be scaled up, then you automatically bring down the cost. So that's one way we call it as a gene agnostic approach, where irrespective of what mutation is there, you can apply the technology. So the process is the same, and you make large batch. So the moment you make a large batch, then it is applicable to a large number of patients in the same batch preparations. So that is one way of cost cutting. Another is having the technology, indigenous technologies developed. So we are now at the early stages. India has started this very late, and we have in the last 5-6 years, we have made good progress. And with more and more such indigenous technologies coming up and our own IP being built up, this IP cost also will bring down the cost, because we are not dependent on any other outside technology. We are building up our own technology for that process. So, but with respect to the tailored therapies, where you really don't have an option, because many of these point mutations are, so it may be the same gene, but the point mutations may be different in one patient to other patients. So it is definitely tailored. But what we are looking at is work out a process that can be broadly applied, and that process we just have to change a small aspect of the editing construct so that you can make it tailored for the patient. So the process is going to be small and it's going to be short. It's not a scaled up preparation, it's going to be small scale preparation. That way we can bring down the cost. And the other thing is the cost is because we are right now relying on most of the reagents which you use in the lab are imported. Imported not only delays, and also you have the added custom duties. We have really failed to invest in developing those consumables locally in the country. So I think even that has to be improved. If that improves and we have the local chemicals and reagents, and even that will bring down the cost. So we are hoping that down the line all this will make this affordable for people.
Thank you. In the interest of time, we'll take only one more question. I know a couple of you also had questions, but I'll request you to take it offline.
For a patient trying to seek these kinds of activities, interventions, what would the usual workflow be, and what would be the expected timelines? I understand there are no usuals in such scenarios, but broadly speaking, what can a patient expect? Thank you.
So you're talking about like a completely tailor-made bespoke therapy for an N-of-1 trial. So I would have given a different answer 3 years ago, but this FENIB trial actually taught us that if we put ourselves into it, it takes about a year, literally. But if it's a if it's meant for an N of 1 trial. The regulatory aspect of it depends on the regulatory status of those technologies at that given point. One of the benefits we had was that we had a facility that was already approved by the regulatory, so that makes it faster. But if you are doing it at a center which doesn't have the facility approved, then the facility approval alone can take up to a year, which is in addition to whatever experimentally you're doing.
Something about the process, means how does a patient really approach you? Means just walk into your clinics and everything happens, or what?
Well, thus far, that's what patients have done. They have come, sat at my office, particularly for the DMD patients work that we do for Duchenne muscular dystrophy. I have to appreciate these mothers who literally sat there as a dharna, that until you start work on this, we are not leaving. And that's how the work got started. And the patient group only supported the research initially. And now, of course, we have broader support to do those work. But that's essentially how it works. It's still not a very formal thing. People just write. I think I get a request like that almost every 2 or 3 days.
Thank you very much, and especially thank you to Arkasubhra and Indumathi.
Can I add one?
Yeah, please.
So what is important is to know if it is a genetic condition or not. I think if the patient should understand if there is a disease running in the family, right? So that itself indicates there is an inherited situation, and you talk to your doctors and figure out if there is a requirement for genetic testing, it can be done. And once you know there is a gene involvement, then the points which Arka told will apply. And so that's it. So it's important to know if it is a genetic condition or not. That's more important.
Yeah.
And not all genes can be treated again, though we say the technology is there and we can treat it and therapy is down the line. But again, not all diseases can be treated even if it is inherited. Okay, so that's the understanding which the clinician would know which are the conditions that can benefit from this approach.
Thank you very much for being this wonderful audience, and thank you very much. This was an extremely informative session.